Sometimes we get a hold of somebody's personal story and it's just got to be shared. And our whole organization got together not too long ago and saw a video about a patient story.
My whole life, I always wanted a child. When Roy finally was born in December 2015, I felt like my life was
And it was so moving that we went and we chased it down further. And we want you, our listeners on Discovery Matters, to meet Roy and hear his story and why it is important in science.
This is the sound of Roy and his mother Maja. Roy's 4 years old and he has spinal muscular atrophy, or SMA for short.
Well, that's devastating. Kids with SMA didn't used to live for more than 1 year.
Yeah, it's awful. So let's go out to the suburbs of Uppsala in Sweden and talk to Roy's parents, Maja Ödmann and Hugo Muhrbeck. And let's hear Roy's story. It twisted, it turned, and as you listen, it becomes really clear that there's a lot to learn about the kinds of drugs that have been made in order to help people like Roy. Okay, so 4 years ago, Hugo and Maja brought home their first child, and like every new parent, they were excited and dazed.
Amen. Do you remember those days?
I do remember.
Terrifying,
it's terrifying,
exciting.
Everybody tells you about the birth and all that kind of stuff. Nobody tells you about like the rest of their lives and how horrifyingly difficult it is to...
I remember actually watching our sons breathe and just like, are they inhaling and exhaling? Isn't it amazing?
And please don't forget to do the second part, because you have to do it again and again and again.
Exactly. So Roy and Hugo, I guess, were like this.
Exactly. And do you remember waking them up just to make sure that
they
were still
alive? Totally.
He's sleeping, but I'm going to wake him up. Oh, I know. It's awful. And terrifying and beautiful. Anyway, so Hugo and Maja brought Roy home, and after a while they started to think that maybe everything wasn't exactly how it should be.
It felt like he was developing slow when it comes to movement and sitting up and crawling and stuff like that.
So they took Roy to their local clinic.
And they just said, you know, all the kids are different, they develop in different speed. So, yeah, we just bought it.
Well, yeah, I mean, they realized something was very wrong when Roy couldn't really eat properly anymore. So back to the clinic they went, and this time they were told to go straight to the hospital.
I think it was 3 doctors that looked at him, and they walked out of the room, and then they came back to Maja and said,
We suspect that he might have spinal muscular atrophy.
The doctor said there is no cure, because this is like a genetic disease that you can't cure. I mean, you can treat it and make things better, but you can't cure it. And I don't know if it was the same day or like the next meeting with the doctor when he said, yeah, I need to tell you, there is a study where they are testing this new medicine. And at that point we were like, okay, when can we join the study? And he said, no, it's too late, it's closed. They won't accept any more participants.
After a week or so, you know, the same doctor rang Maja and Hugo with just unbelievable news. It seemed that there was space for Roy in the clinical trial.
And that was, you know, the feeling was great. But then they told us that there's also a placebo group in the study. So 1 out of 3 kids gets placebo and 2 gets the real thing. And that was kind of a weird lottery to be in. So when we started with the trial, we watched him so closely, everything he did, everything he was trying to do, and we looked at him and noticed change. Did he do that before? I don't think he did. Oh, he can move his legs. He can do that? I haven't seen that before.
We analyzed him so closely when he came back from that room where he got the injection. Like, is he sad? Has he red eyes? Has he been crying? Maybe because the syringe hurt very much, or maybe because the injection was a real one.
Yeah,
So
because
that's
the
why
placebo-
he's been crying. Now he's sleeping. Maybe because he's been crying so much. It's like we analyzed it.
So just so I have a good picture in my head, how is this treatment, whether it was placebo or the experimental therapy, because that's the way clinical trials work, is you have to have a control group. How was that therapy administered?
So this is the thing. If you can imagine an infant, it's administered directly into Roy's spine. Every patient feels a little prick on their back, but no one knew who was getting the placebo and who wasn't.
In August, I remember I was there and I was like, oh, now he's going to meet the physiotherapist and he's going to show her that he's been stronger now because of the medicine. And I remember that moment, he couldn't continue the exercises. Nej, nej, kom igen. Kom igen!
Hjälp, akut! Fan!
Jag får nog hosta. And he couldn't breathe and it was chaotic and I was sweating so much and he couldn't even breathe through this exercise. And I thought he would show that he's been stronger. So that is how much I wanted this, how much I imagined this. And I understand that's why they have placebo in some kind of studies. Not this one, because I couldn't, even my hope couldn't change his muscles or his DNA. But I thought he got it because I wanted it. But then I understood that it was a lost race, that he didn't get it. He was only getting weaker and weaker and weaker, and we were spending more and more time at the hospital, and we noticed that he's not getting stronger, only weaker, but it took months.
So they started noticing this improvements, or Maja and Hugo were looking at Roy and thinking, oh, he's getting better.
That was just in our heads. It wasn't real.
Oh, no.
I know, but don't despair. A few months after they learned that Roy had been on the placebo all this time, the doctors came to Hugo and Maja and said,
There is so much clinical evidence for this medicine to work.
They called us and they said, now we're going into phase 3. Now everybody, including the one in the placebo, will get the drug.
Phew. Oh, great.
Yeah, but hold on. I said this story twisted and turned. It turns out that they would have to wait for 2 whole months for the real drug. And this, of course, the 2 longest months of their lives.
Those 2 months, it was a race against the time, because I could feel and I could see Roy losing abilities every day. I could see, oh, today he can't do that anymore, or today he can't do that anymore, or now he's losing something that he will not get back. He will not gain it back, because that's what the doctors told us, that whatever he's losing, he will not gain back. He will only stabilize where he is.
That is excruciating. So did they get the therapy in the end?
Well, that was a whole journey in itself.
So we took the ambulance to the airport where we boarded the smallest propeller plane. You couldn't even stand up in it. It took us to Gothenburg because that's where this study was. This is also where we had to go every 4 months for the injections. It was a tough trip for all of us, but it was also kind of a journey. So it was mixed emotions.
Okay, I want to hear much more about Roy, but I think we need to get some science at this point. Can you tell me a little bit more about this therapy that they'd waited so long for and traveled so far to get?
Yeah, well, it's an unusual type of therapy that uses something called an antisense oligonucleotide. I've been practicing, so it's an antisense oligonucleotide. And it's better that we get the expert view to tell us some more.
So oligonucleotides are really just short strands of DNA or RNA. In our cells, they're used to regulate gene expression. But of course, they can be synthetically produced as well. And they're used broadly. So think about things like CRISPR, polymerase chain reaction, cloning, molecular probes, forensics, microarrays, They're sort of ubiquitous genomics research. To give you an example, they're used in high school labs, so very broad-based. At Ionis, we use chemically modified RNAs known as antisense oligonucleotides to target RNA.
So this is Max Moore, and he works with these lab-made DNA or RNA strands called oligonucleotides, or oligos for short.
So these are really useful in many contexts. So it's not exactly that they set out to use oligos in therapy specifically. Is that right?
Precisely. I mean, so much so, more than likely, if you're living in a developed area, your life has somehow been touched by oligonucleotides. You know, they're used in forensics, they're used in cloning, they're used in basic genomics. You might have used them as some sort of diagnosis at your doctor's. They might have been used at the laboratory doing the testing. But absolutely, they're used quite broadly. At Ionis, we're talking about chemically modified RNAs known as antisense oligonucleotides that bind precisely to their target RNA through Watson-base-Crick binding. So most people know that term, right? It's been something that our eukaryotic cells have used for hundreds of millions of years. And depending on those chemical modifications, we can do a number of things. So, for example, we can downregulate proteins through an RNase H pathway. We can upregulate proteins by directing splicing, or we can simply degrade a toxic RNA. And that's just, a few examples. But if we kind of go back to the simplicity, I think the way to think about it is the antisense oligonucleotide, which is a synthetic RNA, is the drug, and RNA is now the druggable target.
So why do we hear so little about these miracle molecules?
So the story of how Max got into working with oligos goes back all the way to 1985 when the founder, Stan Crook, was working at SmithKline.
He comes in to SmithKline about the time that Tagamet hits the market, and Tagamet's the first billion-dollar drug, and he sees that this profound change in SmithKline, and appropriately so. You're moving more towards cells and this sort of thing, and Stan is a purist in terms of research, and he sees this small molecule approach. So when we think of small molecules, think of like ibuprofen, aspirin, Lipitor, benzodiazepines like Valium, for example. And that's really what SmithKline is focusing on. That's what Tagamet is. And the challenge with small molecule approach, and it works really nicely, you don't learn anything from one molecule that you can apply to the next. As a matter of fact, even with one molecule, if you add or subtract, let's say, a handful of atoms, you can take a compound that now has a therapeutic effect that's pretty safe and make it where it has no effect, or even worse, make it incredibly toxic. Stan at the time, is thinking, what the industry really needs to pursue is something disruptive, something where, we can learn from each drug that's produced and across an entire platform and have something, that's truly kind of revolutionary in nature. And, he's kind of having these thoughts, obviously, probably quite a bit of introspective thoughts. And at some point, he goes to, I believe, a symposium, and he hears about antisense oligonucleotides. And, I think a light bulb comes in his head, and that's when he sets off in 1987 to found Ionis. Now, what's truly interesting about this is, keep in mind, he's taken a handful of scientists, incredibly bright people. He's working essentially out of a garage in Carlsbad, where literally the labs are separated by visqueen, plastic. And he's saying, hey, we're going to found a genomics-based company. But keep in mind that the Human Genome Project didn't start until 1993 and didn't finish until 2003. What does that mean? It means everything, this is kind of a moonshot approach. We have to build everything from the ground up. Today, you can order, for $100, you can get your dog's genome sequenced. There's nothing like that around. You have, going back to the Human Genome Project, that was a $3 billion project that I think something like 10 to 15 different nations worked on. College students across college campuses everywhere contributed to it. It's a massive undertaking, and here you have just a small group of scientists saying, oh, we're going to be a genomics-based company. So getting back to this grounds-up approach, I can tell you from the manufacturing perspective, a good example. When I started here 23 years ago, I remember, and it was shortly after I started, fairly new, and I'm sitting in a meeting, and we're talking about potential for commercializing a drug that we're working on. It's a cancer drug. And, we're somebody's got their calculator out and they're saying, well, if this many people take this drug, we're going to need this many kilograms of drug. And, finally somebody speaks up and says, I don't think we're going to have enough salmon sperm to supply the market.
Did I just hear him say salmon sperm?
He did say salmon sperm, and he started laughing to himself, thinking that they were just joking.
But what it turns out is, again, we're making synthetic RNA and DNA, but we have to start with adenine, cysteine, guanine, kind of the basis of life, so to speak. They're all being, at this point, they're being derived from salmon sperm. So, you get the salmon sperm, you chemically digest it, you go through some separation process where you can individually separate out the bases, and then there's some chemical iterations and modifications that occur after that. But, they were quite serious. You start calculating, how much salmon sperm you need, and you realize, you're going to wipe out an entire species, potentially a couple of biomes, just to supply the market. And obviously those things aren't acceptable.
So where does Max see this field going in the next 5 to 10 years?
We'll continue to invest heavily in genomics-related tools. We'll invest in all the portions of the platform. Expect to see new routes of delivery. And really what we're talking about here is increasing therapeutic index. So convenience to the patient. We're talking about more potent compounds. And what I think is really exciting personally is this sort of LICA technology where, we'll be able to direct drugs rapidly, one of them, at least actually two of them, in phase 3 compounds using a LICA, where we can direct the drugs to specific organs and, more importantly, specific cells. So if you think about it kind of in layman's terms of what's going on here, is if you were to cut yourself on your finger and you didn't take good care of it, or maybe you're having a wonderful vacation in the tropics and you suddenly have an infection, you're going to go to the doctor, they're going to give you an antibiotic, and you're going to have to take it for a while, and you take it, it gets distributed across your organs and in your blood, and eventually a small amount of it makes to that point of infection. Because if you have to take such large amounts, you're probably going to have to experience these side effects, things like, intestinal discomfort, because your flora gets completely wiped out. And, you're going to have to cut your vacation short because you can't be in the sun. The list goes on and on from there. But imagine for a second if you could take just a very small amount of that same antibiotic, and when you took it, it went to the cells that were infected. So that's kind of what we're talking about with LICA technology, is being able to direct the drug exactly to its point of action.
And for Max, this work is just fascinating, it's personal, it's rewarding.
A lot of people do research and they never get to see, their discoveries broadly applied. But for a manufacturing person, to be able to do research in a lab and then see that actual discovery applied to a manufacturing process for a drug that's going to go into a toxicology study with animals and then immediately into human beings, that's pretty exciting stuff. Here at Ionis, we have this tagline, sick people depend on us. I really live that, and then we really live that. There are a few things as humans we share in common. On one end of the spectrum you have birth, and on the other one you have death. And everything in between gets really gray. But more than likely, unless you're just incredibly lucky, you're going to get sick. For me, that's the most important part of my job, is knowing that I have a hand in potentially helping people along that are sick and helping them out.
Yeah, Max is super inspiring. He knows all of these patients that he's making a difference for. And we're learning so much here that I think we definitely need a second chapter on this type of therapy and more patients. What do you think?
Yes, we definitely should. But first, let's check back in with Roy.
Today Roy's 4 years old. He's going to daycare with his assistants. And he's a happy boy. He likes to play. He just started to play memory and he loves it. Every day he wants to go downtown and look at trains and buses. Every day. No matter what time it is, if it's in the middle of the night. He still can't sit up. He can lay down and he can almost turn him over to the other side. The improvement is really, really slow right now, but it is there.
I remember between the injections at the beginning, I could see one improvement, but that's because we started from zero. Roy could only move his eyes when we started. So after the first injection, I could see him moving the shoulders, and then after second I could see him lift his underarm. After third, I think moving his head from side to side. So I could see one thing every injection in the beginning. But now he's just improving what he already gained. Like now he's moving the head very fast. So now he's lifting the arm very strongly, but he's not lifting the upper arm still. He's getting stronger in another way now.
But the reality is that Roy is looking at receiving this therapy or dealing with spinal muscular atrophy for the rest of his life. And so on a future episode, we're going to meet another parent of another patient who has been receiving this therapy for 5 years already. And we're going to talk to the physician who is running that long-term clinical trial.
Until then, thank you for listening. Rate us on your favorite podcast app, and we'll see you next time.
Bye for now.